You have fallen into Event Horizon with John Michael Godier. In today's episode, John is joined by Stephen Bennu. Stephen Bennu heads the Foundation for Applied Molecular Evolution, which he founded after serving on faculty at Harvard, ETH Zurich, and the University of Florida. His research combines two traditions in science, one from natural history and the other from physical sciences. His laboratory has been a leader in the field of synthetic biology, where it has redesigned DNA to better understand how these molecules work, generate new classes of diagnostics tools, and to open new avenues to disease therapy. Separately, he also created the field of paleogenetics. His work also guides NASA missions to search for alien life and how life originated. His most recent book is titled Life, the Universe, and the Scientific Method.
Dr. Stephen Bennu, welcome back to the program.
Well, thank you.
Doctor, we have a very exciting finding regarding the asteroid Bennu, and NASA returned a sample of that in 2023, and now the analyses of these samples is coming out. And there were 3 papers that were announced, and one of them was very interesting in that it finally sealed the gap, the sugar gap of detecting ribose in this case, and a bunch of other sugars, including glucose, in this asteroid, which was the last building block for RNA in space, proving that this could have happened on early Earth, whether delivered by an asteroid or just Earth had it. So we have the materials needed for RNA. Can you give us a rundown of what this finding means and why it's important?
So what you just said here is you're filling in the gap, and this is a model of RNA. Of course, it's not quite so pretty colored in real life. But what you have is this so-called double helix, which is the backbone that is being traced in white that wraps around itself. And it's sort of a ladder. So you go up the steps. The bases are the things that hold the 2 strands together, and those are what carry the information. They come in 4 kinds in your body. They go by the letters A, G, U, and C, which are adenine, guanine, uracil, and cytosine, but that's not important. What is important is that we've known how to make those bases for maybe 50 or 60 years in a prebiotic atmosphere on early Earth in certain kinds of environments. And that's been shown in the laboratory. That's an important demonstration because it's in the laboratory that we can study actually how they're made. But it's even also important is that they have been found in what are called carbonaceous chondrites. These are meteorites that come to Earth. Unlike stony meteorites or iron meteorites, these contain a lot of carbon in them. And the asteroids, like Bennu, is an example of a carbon organic molecule rich and meteorite or asteroid out there in space. And so this is important because carbon is essential to life as we know it in any case. And the fact that you can get these bases in the RNA is actually quite important because that means you can get these bases without life in the bases that are needed for RNA. That means that you have natural processes that can deliver those bases to early chemistry on Earth that would lead to the first RNA, which is thought to be the first molecule, genetic molecule, the first informational molecule to support life. The second thing is these backbone white things. These are phosphates. They form the back. That has also been known to be out there in space, here on Earth, in rocks all over the place. But the reason why this result is exciting is that they have found on asteroids. Bennu is the name of the asteroid that, as you mentioned, they brought samples back a couple years ago, which is these red things, and these are sugars. They're sugars like glucose, and they did find glucose, a very common sugar in your metabolism, in the asteroid sample that they brought back. This glucose has 6 carbons in it, and you burn that for metabolism. Ribose, which is a different kind of sugar, but it has only 5 carbons, but it is used to hook together the RNA. These sugars are rather unstable compared to other pieces of this molecule. So if you go into the kitchen, for example, and you heat sugar in water for any period of time, it will form caramel. It will caramelize. It turns brown, and it's tasty brown. So as long as you don't heat it too much, eventually it will turn black. So one of the major questions that you've had for years is, how do you get a hold of these red units to build the strand of the double helix when those red units are ribose, and ribose is unstable? And so there's been a major question as to how that happens. Now, we've gotten involved in this maybe 25 years ago by pointing out that certain minerals, and I'll put this down in the show and tell and pull up one of them. This is a mineral that contains a lot of borate. Sometimes you can buy this in museum gift shops. It's called TV stone. If you polish this, it's sort of nature's fiber optics, so you can actually see light be transmitted. But this is a boron rock from Death Valley, actually, where borax is concentrated. You know borax if you have used this as a cleaning solution in your laundry. But the bottom line is that boron will grab a hold of the ribose, the red sugars, and stabilize them. And that's been a very important part of this process for how we could get laboratories to make a laboratory synthesis to make stable ribose under simulation for prebiotic conditions. But what's really exciting about this result is that we got out of the laboratory. You never know, right, when we can get something made in the laboratory. That's just because we have our good hands and we're making sure that the right chemistry happens, the right reaction occurs. But finding this out there in outer space says that, yep, that kind of process can also occur naturally without my hands guiding and making sure that things turn out okay.
I have an off-the-wall question for you. In my deep dive on these findings and reading the papers and digging ever further as I do you think that fructose can occur in space? Or does that have to either be the lab or a fruit?
Yeah, no. It's very interesting. So keep in mind that fructose has 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms, just like glucose. Therefore, the chemists will call them isomers. They have the same masses of atoms. And in fact, you interconvert in your body in metabolism glucose to fructose, and that's one of the processes by which you burn glucose to get energy. But the most stable arrangement of atoms with 6 carbons, 12 hydrogens, and 6 oxygens is glucose. So that means if you set the system up and you let it come to what the chemists like to call equilibrium, where there's no more energy in the system, but you have a certain amount of glucose and a certain amount of fructose, there's more glucose than fructose. And so what happens when you want to get something called high fructose corn syrup, right? They take corn from Iowa, they put an enzyme in which catalyzes interconversion. They get about 65%, well, maybe 55% glucose, 45% fructose. So yes, they can get it to work, but the fructose is always number 2 in that pecking order. But the answer is sure, they can go look for it. Keep in mind that this is a sample that has gotten at great expense from an asteroid that's zipping through space without waiting around for us to land on it. And so the amount of material they actually bring back is very, very small. So it's quite a challenge, and this is one of the reasons why Yoshi Furukawa, who's the first author on this paper that you mentioned, is such spectacular work. They had to be able to do very careful analysis of very small amounts of sample to find the sugars that are in there. But that's part of the process. So yes, fructose might be there, but they might not be able to find it because there's so little of it. But ribose is, a compound that we expect to have in large amounts, especially if there's boron, and especially if there's minerals like what we get from Death Valley in that rock as well.
But, and here is what interested me the most because of the implications of it. They did not find deoxyribose. The Furukawa and colleagues did not find that. And that is what you need for DNA as opposed to RNA.
Yep.
This lends credence to a very, at this point, somewhat old hypothesis of the RNA-world, where the beginnings of life actually start with RNA. Now, there were always spooky things regarding this because we have things like RNA viruses, including a certain pandemic, so RNA is important, but RNA is rather fragile. compared to DNA. And I remember some people were saying, well, maybe it could polymerize with itself and maybe hold itself together better somehow chemically and all this. And what are your views on this strengthening the RNA world hypothesis as the first step of life on Earth?
Right, yeah. So exactly. I mean, I'm a big fan of the RNA world, as are you. So we are able to make, well, we published about 3 years ago a paper. The first author is Craig Jerome, who's the student who did the work, which showed that you could actually take ribose phosphates, nucleoside ribose phosphates, and on a rock called basalt, which is a volcanic rock. I have an example of it here. This is actually a basalt from Arizona. And you can see the green olivine, which is the gemstone peridot here. But if you try to crush this rock up and make basalt glass out of it, something that you now see in active volcanoes on Hawaii or on Iceland. And also, this is a sample here. We actually know that basalt is found on the surface of Mars. In fact, much of the surface of Mars. And the reason we know this is because I have a sample of Mars in my left hand. Now, this is not something that NASA brought back, but this is a sample that we get by having something hit Mars and eject rocks, which then wander around the solar system for a little bit and then land in the Sahara Desert, where the camel herders are smart enough to know that they see a rock on top of a sand dune, it fell there, and there's somebody from the United States who's willing to pay them money for it. So this is how we come to get these things. But these are basalts. And so it turns out that you can actually make RNA molecules, long RNA molecules, and certainly longer than this, right? Maybe 100, 150, 200 building blocks long, which is enough information to actually support Darwinian evolution. And so we think that the RNA would have been the first genetic molecule, the first information molecule, the first molecule that met the information needs of evolution on Earth, and the fact that we can make it in the laboratory in relatively high yields in the presence of boron, right, and the fact that they can find it on asteroid Bennu is quite, it's a convergence of observations that is very auspicious for the RNA first model for the origin of life in the RNA world. Now, deoxyribose doesn't do this. Deoxyribose is not stabilized by borate. It doesn't get formed in laboratory processes. It's not a particularly useful, interesting compound. It does convert itself into caramel if you make some of it. So the story is that you made RNA first. That's how you got Darwinian evolution started. That's where the informational needs of the first life came from RNA. And then what happened is after you got life going, the intrinsic instability, or as you were mentioning, fragility of RNA was recognized, and somebody invented a way of ripping off one of the oxygens off of RNA, which is C5H10O5, to make deoxyribose, which is C5H10O4. Hence deoxy is gone from 5 oxygens to 4 oxygens. And that molecule now provides a much better platform for information storage because it is less fragile. So the idea is that DNA came after RNA. And one of the pieces of evidence for that is that all the DNA building blocks are actually made in your body from RNA building blocks.
To reiterate your point, I too have fragments of Mars, but much smaller. But mine are, this is Nakla, which is the Mars rock that supposedly killed a dog when it fell in Egypt in the early 20th century. Now, speaking of Mars, this has direct implications because obviously with the basalt and everything else, Mars had hot springs. Mars had all of everything it needed. And if it had all of these building blocks, who is to say that, there could have been 2, maybe 3 planets in the star system at varying times early in their history producing RNA chemistry. And even, dare I say, Io. I don't know how much legs that would have.
Well, no, that's a fair point. So what we have published, and there's going to be more coming out in the not too distant future, is a way of getting from very simple molecules, formaldehyde. It's got 1 carbon, 2 hydrogens, 1 oxygen, all the way to ribose. getting reduced compounds like adenine, guanine, A, G, C, and U, and phosphate. And we have a process that goes all the way from small molecules all the way through to RNA on these basalt rocks. And so the idea that this happened on Earth, well, Earth is not the only planet that has basalt rocks and the conditions for doing this chemistry. Venus probably also had these conditions. It's just that Venus is a little too close to the sun, and it's got a little hot and had a runaway greenhouse effect, which means that their atmosphere is sulfuric acid, which is not very conducive to RNA. But Mars, okay, is a little bit too far away from the sun. It's a little bit colder, and of course, its accretion was disrupted by Jupiter, so it didn't get to be quite so large as Earth. And so it does not have the atmosphere that you would really like it to have. But there's no reason why 3.5 billion years ago, when there was an atmosphere and there was water on Mars, and you can see the remnants of that water, that you would not have also had RNA being formed. And I don't know whether you followed, there was some work, the first author of this paper is Hurwitz, and they found, and again I have props which I neglected to bring with me, but they found what looks like a mineral called iron phosphate. The mineral is called Vivianite for no particular reason, except that the man who found it was named Vivian. But the bottom line is that this Vivianite is the kind of thing that you see crystallizing out of decaying living matter on Earth, where you've got a lot of iron and a lot of phosphate, and the growth of these very nice crystals is started by the organic material to what we call nucleate the growth of crystals. So they have seen specks of this, and they published this, I guess it was in September or maybe October, but they have, and the preprint was earlier, so we know that it's out there, but they have seen on Mars in 3.5 billion year old sediments, give or take, in an environment where there was running water, these are water deposits, they have seen what we would recognize on Earth as the residuals of living systems. Now, you can get this in Earth. We have great shells from mollusks that died, and then they grew these nice iron phosphate crystals in them from the excess iron and the excess phosphorus on Earth. The things on Mars are much smaller, so there's going to be an argument about whether they're the signs of life, but I find that to be an extremely useful biosignature.
It was very exciting. That was a Mars rover finding. We actually did a show on it with a scientist, because the Vivianite was somewhat telling that, yeah, we need to look a little bit closer here with the sample return mission.
Well, in the NASA culture, yeah, I mean, I was just going, we just about to publish a paper on the Viking mistakes and how they perturbed Mars exploration for a half a century. And in the NASA culture, you don't actually say to the audience that you may have found biological material. The culture is very much, you're required to say, we maybe have maybe a hint of what. So you're never allowed to say forthrightly to the public what we've seen, our best guess, good faith statement about what it means and why that statement might be wrong. I mean, this is what the public wants, right? What NASA culture does is you have to be very cautious about interpreting results. Now, I'm not sure what caution means in this context. It means that you walk around the laboratory in your stocking feet, being very careful not to make a sound. It's not clear, right? In principle, the good scientific method is that when you think you've discovered something that indicates life on Mars, what you're supposed to do is play Ender's Game, right? I'm sorry, hard to bring an Orson Scott Card reference in here, but you're supposed to immediately pick the opposite side as your own, adopt the opposing point of view, and of course you adopt it like you mean it so well that you come to love it. So there's the Ender's philosophy. But the idea is that's not caution. What that is establishing internally both sides of the argument. And of course when you do that, it's not only that you help work things out, but you actually understand by that process what the argument is, what you're arguing about. And it really helps to have both sides of this argument laid out. What happened in Viking in 1976 is they misinterpreted some results about no organic molecules on the surface of Mars. We know those results are wrong. The interpretation is wrong in any case, and so that basically shut down the discussion. So no one actually had the science back and forth. So a couple of weeks ago, we published an e-letter in Science magazine correcting a mistake in a 1976 science paper, which started this whole story that Mars has a very strongly oxidizing surface that does not allow life to exist, and therefore Mars's surface is self-sterilizing, and therefore there's no reason to go looking for life on Mars. In point of fact, all of the Viking results were positive for life under the conditions in which the experiments were designed, and by a preponderance of evidence standard, as we say, it's more likely than not that Viking found evidence for life on Mars in 1976. But the way the culture works, it got sidetracked, sidelined, and buried. It's a fascinating story of how sociology works in science.
Well, I was the last person that interviewed Dr. Patricia Ann Stratt, who was the lead scientist on the labeled release experiment. And she went to her grave believing, I found it. And she laid out a very convincing argument to me that Mars appears to be alive. And there's other weird things going on, like methane blooms and all these other little hints here and there at Mars, and even the upper cloud decks of Venus, that suggest if we're going to go there and send people there, we need to know about this first.
Yeah, no.
Absolutely. We need a life finder mission, a direct life finder, which, as you say, NASA's shy about, but people always say that, have conspiracy theories. Oh, NASA's never going to tell us. But NASA is the only organization in the world that has announced the discovery of alien life twice, and was called into question both times. But the point of fact is that you're not the only scientist re-examining the Viking results, because it's just needs a re-examination and it needs a repeat. But do you think we could do it better? And do you think, and here's the next step that will get into all of this, do you think we can determine what chirality that organism might be with a remote experiment?
Yeah. So Gil also went to his grave. He was 97 when he passed away about 3 years ago. He also was persuaded that he had found life. He wanted to send an experiment to Mars to look at the degradation of left-handed food and right-handed food. In 1976, the budget and the technology was not able to support that mission, so they sent a mixture of the two. The food was radioactively labeled with carbon-14, which is a radioactive isotope, and so if I feed you radioactively labeled carbon-14 food into you, you will exhale carbon dioxide that's radioactively labeled, and they saw it. And the problem was that it would have been a much clearer biological definition if they saw the left-handed food was converted to carbon dioxide faster than the right-handed food, which is what happens in your body. Actually, it's the other way around, but never mind. But they didn't have the money for it. So years later, decades later, Russia tried to launch that experiment. And that experiment and the launch ended up in the Pacific Ocean because the launch failed. So for years, NASA, in my view, didn't respect Gil sufficiently, right? He was treated sort of as a crackpot. He was no fool. I got to know him quite well, but the bottom line is that NASA was burned by the 1976 results, which they thought was a problem because the results were ambiguous. I mean, after the Viking, GC mass spec is the instrument that did it, the gas chromatography mass spectrometer allegedly found that the soil was strongly oxidizing. Gerald Soffin, who was the program scientist, said, that's the ball game. No organics, no life. And within weeks, the labs were closed, people were fired or moved on to other things. And of course, nothing in government ever shuts down, right? So to have something shut down is quite traumatic. And so, and if you look at the record for 20 years, nothing flew to Mars. And it was only because, as you mentioned, the second time they announced life on Mars, which was Dave McKay, having observed, and again, in some of these meteors like the Nakla, that you have structures that maybe look like cells, we got Mars back on the menu. But it's funny. I mean, I don't know how detailed. It's not a complicated discussion. They did not know that the Martian soil contains perchlorate. That was not discovered until 2009. The gas chromatography mass spectrometer was the instrument that they were using to try to find organic molecules in the soil. So what they did was they scooped up some soil, they put it into a little bucket, they heated it up to 500 degrees centigrade to blow the organics into a stream of gas where they could separate them and analyze them. But not knowing that there was perchlorate there, they did not know that the perchlorate, when you heat perchlorate with organics at 500 degrees centigrade, you set them on fire. This is fireworks. So what they did basically was, before they had a chance to look at them with their instrument, they burned them, okay? And so that's a problem. Now, they did actually detect some organics, methyl chloride. They detected it in that experiment. And it turns out that methyl chloride is a product of heating organics with perchlorate. And Rafael Navarro Gonzalez, a nice Mexican guy, pointed this out in 2009 after they found perchlorate on Mars. As a human touch, he died of COVID actually a couple years ago. So Rafael pointed out that when you had organics on Mars and you had perchlorate with them, you heated them like the instrument did on the Viking, and you make methyl chloride. So Navarro Gonzalez basically explained all the Viking results not as the statement that the soil contains a strong reactive oxidant, but it contains perchlorate, which becomes an oxidant when you heat the damn thing up to 500 degrees centigrade and make a fireworks out of it. But that never got internalized. I mean, Danny Glavin knows it. The Curiosity rovers have fought this because the Curiosity rovers also have chemistry trying to detect organics on Mars. They encountered this problem. They see the methyl chloride, and what they've now done is basically MacGyvered it, right? They've basically taken that instrument, which was not designed to find organics in the presence of perchlorate, but they have done stepwise heating, and they've managed to find organics nevertheless. A lot of them are chlorinated because of the perchlorate. But what happened was very amusing, was that when Klaus Biemann, who's a professor at MIT, very nice guy, he was the guy who built the instrument to detect organics on Mars. He wrote an article which had all the data. He said, we detected methyl chloride on the surface of Mars. We did not detect it when we tried the instrument on the flight out to Mars. By the way, they also detected freons in the surface of Mars, but they did detect those in the flight out from Mars. So those freons are contaminants from Earth. But the methyl chloride was not, and Biemann pointed this out. So he says in his original paper, we don't know where it comes from, but you know, it could be indigenous to Mars. I can't figure out how it got there. But when he wrote the science paper, which is the review, he managed to put methyl chloride in a table under the heading terrestrial contaminants. So if you go to the original Science 1976 paper, you'll now see a note from us saying that that was a mistake. But then you got a guy named Norm Horowitz who actually designed the photosynthesis experiments for Mars. He put radioactive carbon dioxide on to make sugars, the opposite of what Gil Levin did, which is put sugars on to make carbon dioxide, respiration versus photosynthesis. But, you know, he tried to review the review, and he says, well, all that they found on Mars was cleaning solvents. Well, he's a biologist, he's not a chemist. Methyl chloride, he's confusing it with, you know, chloroethane, which is something that you go to your dry cleaners and get. Methyl chloride is a gas. It boils at minus 24 degrees centigrade. Nobody uses it as a cleaning solvent. But since Norm is a biologist and does not know what methyl chloride is, doesn't know that it's a gas, and has confused it with what he knows from the dry cleaners, he writes that this is a cleaning solvent that they discovered. And then you look at the review of the review of the review. It's like playing, what was this game, a party game called, I think, Telephone or something like that, where you whisper something to the guy next to you who whispers to the guy next to you, and then at the end of the cycle you discover how much it's been distorted. So you have in 2022, just a couple years ago, a NASA endorsed document from a workshop which was staffed by the creme de la creme. I mean, you had to apply to get into this workshop. They had like 200 applicants. They accepted 80 people. Vicki Meadows and Heather Graham. Vicki's a very well-known astronomer at University of Washington. Heather is at Goddard Space Center near D.C. They basically don't know the facts, and they actually think that this soil of ours is strongly oxidizing, even though it's not there at all. But then what's kind of fun is to watch this. What happens? What do you do whenever you get a result in the lab that disagrees with your hypothesis? Well, you're supposed to have one ugly fact throw out a beautiful theory, but that's not what happens. What you do is you brush the observation under the table, hoping that it will go away. So Horowitz actually tried to fix carbon dioxide. Okay, so what did Norm do? He put carbon dioxide onto the soil, right? If you do this for plants outside, the soil will fix carbon dioxide. Okay. And then the problem was that he put radioactive carbon dioxide in, so he now has radioactive soil. So what does he do? Well, the first thing he does is he blows away the excess carbon dioxide so you don't have this radioactive carbon dioxide sitting there to confuse you. But then to get the organics out of the soil, what does he do? He heats the soil to 635 degrees centigrade. Now, he's not thinking about perchlorate. But he observes, and you can see it in his writing, he says, well, we only got about 1% of the radioactivity to absorb on a material that was designed to capture the fragments of the organic molecules that had been fixed in the soil by photosynthesis. 99% of it, for some reason, came out as carbon dioxide. So he has observed the perchloride oxidation of the organics that were fixed in the soil. But since he hasn't thought of perchlorate, he says, and he sweeps it under the table, he said, this must have been absorbed to the walls of the container or the soil grains. Never mind that they hadn't seen this before and never seen it since, and the walls of the container don't absorb carbon dioxide. What he's looking at is the burning of organics on the soil as he's trying to get the organics out of the soil, just like Beeman did with his mass spectrometer gas chromatography, where he heated it only to 500 degrees. Like, we're surprised that he got anything onto the organic fragments out, but he probably got some methyl chloride. There you go. So Horowitz sees the data, it contradicts his theory, and then so Norm writes the following statement. You can actually, in the paper, the culture is now accepted is that the surface of Mars is very oxidizing. You touch it, you get your finger burned. You put Gil Levin's food on it, it gets burned to carbon dioxide. The meteoritic organics from carbonaceous contact, they get burned. It's a self-sterilizing soil. That's how they have. And so now Norm writes, it's kind of odd. We saw absorbance fixation of carbon over 5 days. Now, if the soil were so strongly oxidizing, it should have been burned. So how the hell did the carbon photosynthesis allegedly fix carbon on this strongly oxidizing soil? And then what Norm writes is, I'm quoting more or less verbatim, we don't know, but experiments are underway. I'm sure we'll resolve this problem, and when we do, we will understand also the GC mass spec results. Until that happens, there's still a small possibility. It's actually, he starts the phrase by saying it's hard to explain this without life. Okay, so his closing sentence is that until this happens, until experiments are done, we won't be able to rule out life. Now, we teach students never trust the outcome of experiments that have not yet been run. Okay, but what he has done is basically said that he can explain away this anomaly, which is God speaking to you, right? When the experiments aren't doing what you're expecting them to do, you're supposed to say, it's trying to tell me something, But Norm basically called methyl chloride, which boils at minus 24 degrees centigrade, a cleaning solvent, observed the fixation of carbon, burned it off, didn't understand that it was what was happening. None of us did. And then he said, well, it must be absorption onto the walls of the tube, and moved on. Okay, and the textbooks moved on. If you go to Wikipedia, they will, under Norm Horowitz's page, he's got a nice Wikipedia page, he will say he's the first person to get results that showed there was no life on Mars. No, he's the guy who got the first results that showed there was life on Mars. And by the way, if you go back and do the calculation and you say, wait a minute, I'm burning most of the fixed carbon. How much carbon did I fix? Well, the answer is about 100 cells per cc, cubic centimeter of soil, which is roughly quite compatible with what we see in Antarctica or the attack in the desert. In very resource limited on Earth, we'll see about 100 microbes. So he's fixing about enough carbon over 5 days by photosynthesis to support maybe 100 cells. And that is a damn good number to be compatible with how much label release Gil Levin saw when he fed radioactive food. So we've actually published on our webpage this idea which explains all these results in terms of a bacterial autotroph, someone doing photosynthesis. There's also oxygen is released, but you got to keep in mind that the Martian bugs have to survive overnight. Your plants survive overnight. When the sun's down, right, the plants don't do photosynthesis. But what they do is they do respire with the oxygen for overnight metabolism, burning the organics that they made during the day. So, but the problem with Mars is there's no oxygen in the atmosphere, so the bacteria will store the oxygen for overnight metabolism. And if you throw the acronym together of a bacterial autotroph respiring with stored oxygen for overnight metabolism, you come with the acronym BARSOOM, bacterial autotroph respiring with stored oxygen for overnight metabolism. And if you're not a big fan of Edgar Rice Burroughs and have never read John Carter or The Princess of Mars, you would not know that the Martians in that book call their planet Barsoom. A coincidence? I think not.
Coincidence? No.
Who knows? No, but the bottom line is that, so when I say that there is by the preponderance of evidence, I mean, you go into a courtroom, right? They can put you in jail if they get proof beyond a reasonable doubt. There is no, of course, if your lawyer is, if the prosecutor is very dramatic, they'll say you're a scumbag. We'll prove it beyond a shadow of a doubt. But if you go to a civil trial, it's a preponderance of evidence, then, more likely than not. And so there's no proof in science. You're never going to get it, and not in the mathematical sense of the term. But but by more preponderance of evidence, it's more likely than not that they found life on Mars. And poor Gil. Gil had read our papers. We started published the first one here about 25 years ago. So Gil said, wait a minute, Benner has shown that the GC mass spec result was misinterpreted, that there is, in fact, the soil is not self-sterilizing. So Gil, for the last 20 years of his life, wanted to know why the hell we didn't go back and to the status quo ante and, re-evaluate the system, send another mission back, right? And the short answer to this question is that once you get this idea embedded in culture, it's very difficult to dislodge it. The problem with science, as you know, is that it is done by scientists who are Homo sapien, ape descendants, who get themselves in a groove and do not let logic, facts, or reasoning overturn what it is we have come to believe in our brains.
I can't tell you how many times I've come across the cleaning solution thing. Still, to this day, it's
really?
Still persisting and saying, well, it was cleaning solution, and it's wrong. It wasn't.
Yeah, no, it's wrong. What's more to the point, not only was it wrong, but if you go to the original papers, which we also teach the students, right? We teach the students that they're supposed to go read the damn paper. Don't read the review of the paper or the review of the review, and certainly don't trust the textbook. But yeah, so in the original paper, it's clear. You don't have to be a rocket scientist here to be able to see what is in that paper. He says it, methyl chloride. It's not seen in the flight out. It's not a terrestrial contaminant. He said we might have expected to see methyl bromide and ethyl chloride. Well, there's no good reason for that. He's trying to find some way to resolve a problem that he knows he has. Beeman knows he has this problem, and he wraps the paragraph by saying that we tend to believe It could be indigenous to Mars, is what he says, but we tend to believe it is not. But the question is, why the editorializing? And we teach students, you can go, it's like reading Shakespeare, or in your English literature class in high school. You can read the sentence and you can see the author of that sentence telling you facts, but also editorializing. Okay? And you can read this text and you can see these people editorializing, and when they're editorializing, whenever you see somebody editorializing in their results, it's some place that you should look more carefully, because they're probably trying to rationalize away something that they don't understand. And that happens all the time.
Yeah, that is a problem.
Well, but it's also a problem that most of the time it should be rationalized away, right? Most of the time your experiment doesn't work because the machine is broken, or you dropped the flask, or there was an impurity in the reagent that you used. I mean, in general, it's very difficult to do science. Science is much too hard for scientists. And so you very often encounter anomalies where you must just ignore them, because they're actually, if you spend every time trying to track down a broken piece of glassware or a defective instrument, you spend your entire life tracking these down. And so, but never mind, when you get to the point, when you understand what's an important anomaly, what's a consequential anomaly, you pursue it. And one of the amusing things about this is you can read these guys' papers. They know that they have an anomaly. They know it is consequential. Right? And they say, oh, I'm sure that some experiment in the future will get rid of it, or maybe the stuff's just absorbed to the glass walls, the sides of the tube. It's really, well, of course, it is what makes science fun, right? I mean, it's not fun. Science is a miserable occupation, but it's like solving the Sunday crossword puzzle, right? That's not fun. But it's certainly an intellectual challenge.
Now, what would that life look like if it's surviving on effectively the surface of Mars, or at least in the soil? Now, another objection people are going to point out is the ultraviolet environment is sterilizing it in its own right, but that only goes not very deep in the soil.
No, but you have to worry about that, because remember, Mars is frequently covered by planet-wide dust storms. So none of this soil has ever been, well, that's not true. I mean, in nooks and crannies, I suppose, but a lot of the soil has been stirred up into the atmosphere, and it has been exposed to ultraviolet light. So, I mean, there are species on Earth that are resistant to ultraviolet light. Now, go outside. That plant also has to survive ultraviolet light. You try it sometime. You go outside. You got to survive ultraviolet light. So one of the strongest, what we call in evolutionary biology, cline, which you have, where I can tell what latitude your DNA was, adapted. And you'll see this cline it's not really a racial thing, right? I mean, in India, there's a cline across the subcontinent. In China, there's a cline across the subcontinent. The sun is poison, and you put things in your skin to try to prevent the sun from killing you, with the trade-off being that you also want to use the sun's energy to make your vitamin D. Go outside of that poor plant. That plant is sitting there with carotenoids, all sorts of things in that plant to try to get rid of the harder side of that ultraviolet light, because that will also toast the plant, even with the ozone. All right. So with Mars, you have to have the same thing. There's something called a cryptoendolith. These are organisms that make their shells out of rocks. And so one of the things that's abundant on Mars relative to other things is rock. So, you might very well expect to have a hard shell formed by mineral precipitation on the outside of these microbes, which is protecting this thing from the harsh ultraviolet light. But you're absolutely right. A second problem that you have is that you have water which is very salty. And on Earth, if you go down the Dead Sea in Israel, the Israeli-Jordan border, right, the north end of it is being fed from the Sea of Galilee, actually, by the Jordan River. And so it's relatively low salt, but by the time you get to the south end, it's evaporated. It's very high concentrated salt, and it's precipitating out. So life on Earth gets down to a point, but doesn't go all the way. to the very bottom end, the south end of the Dead Sea. And the argument has been that there's a certain upper limit to salinity. And the way they actually phrase it is they don't talk about the salt in the water, but they talk about the activity of water. So pure water has an activity of 1. There are people who will tell you that life cannot survive below an activity of water of 0.7 or 0.6, saturated magnesium sulfate, something like that. But of course, again, the logic never works. What they're telling you is that life that we know on Earth does not survive that. And you got to keep in mind that the reason why salt is a problem is not because of the RNA. That's actually does just fine in salt, but it's because of the proteins. And more importantly, the 20 amino acids that you have chosen for your proteins, which are a problem for on high salt. But if I were on Mars, as the planet is cooling and drying, as the water's going away, the salt's going up, I'm going to have amino acids evolve that will be able to form proteins that behave in salty water. And so the problem that you have in Mars is the water is intermittent. The salt is actually kind of useful because it may help suck in moisture from the air. Keep in mind that Viking allegedly saw fog. So you do have allegedly times in the Martian day-night cycle where the relative humidity is 100%. And that is conducive for some level of life. And if you're talking about 100 cells per cubic centimeter, you know, hey, it's what you see in the Atacama Desert.
That's still a lot. You know, that's still a lot.
It's yes, and it's remarkably comparable to A, how much food Gil Levinsohn processed, and B, how much oxygen was released, where the hypothesis is that this oxygen is being stored by those microbes for overnight metabolism.
Well, it would make sense. Your thoughts on Venus and the phosphine?
Yeah, well, you know, you probably haven't read our papers on the subject, but let me just give you the high point. So sure, when Jane Grieve was the professor at the University of Cardiff in Wales, first published phosphine, and said she couldn't think of any way of making it abiologically, therefore there must be biology. I wrote a nice thing on our blog, primordialscoop.org, which said this is crazy. Okay, the clouds of Venus are 95% concentrated sulfuric acid. And then I thought about it, and then, you know, why is it crazy? Well, you know, it's not so clear. Carl Sagan in 1968 had declared authoritatively that you could not have organic molecules. Organic molecules could be absent from Mars. But then Jan Spacek, who is a very bright astrobiologist, got his degree in the Czech Republic, and he's, you know, has the skepticism that a good scientist should have, said, wait a minute, who says organics are not possible in strong sulfuric acid? Of course organics are possible in strong sulfuric acid. And so he made a model for how you could get organics which then would fall. Of course, eventually they become very, very hot. And then they break apart, they become carbonized, and then you have a reaction that will restore carbon dioxide and carbon monoxide in a planetary cycle. And so at that point, I'm a physical organic chemist, as we like to call ourselves. We worry about mechanisms of chemical reactions. So I actually went and helped him in the laboratory. This is great fun, to analyze how you would go from a one carbon compound, CO2, CO, formaldehyde, to a two carbon compound. Once you're at a two carbon compound, you can go on to higher carbon compounds as well. And so we actually showed that we could do this in the Venus atmosphere, and that the two carbon compounds would be stable for a period of time, and they could form long chains. And so the argument is now maybe it's not so crazy to have life in the sulfuric acid clouds above Venus. It just would be a chemistry very different from what you and I have. But then Jan went further. One of the questions is, what color is Venus? Well, the answer is it's a pale yellow. But if you look at beer, right, you look at the froth of the beer, it's pale yellow, but of course the liquid below is dark. And so that's because you're looking at scattered light. And, if I take smoke, depending on how big the particle size, it can be white to black, right? Smoke can be a very, very fine particle. So Jan then managed to get some collaborators which built an entire model for Venus. We have a good idea based on what we can observe what the actual color of Venus is. We had a guy, undergraduate, came and did some rotations here right after he graduated, named Victor Moncada, and Chris Temby also helped, and Gage Owens helped. These are all undergraduates going on their way to graduate school who stopped off in our place for a year, and they found fluorescent molecules in this sulfuric acid model that we did in the laboratory. And now, in a couple of months, they're going to launch a rocket from the Mid-Atlantic rocket launch place in North Carolina and Virginia, actually, or Maryland, actually, but they're going to launch a rocket to Venus to look for the fluorescent compounds that we saw in the laboratory here. So so from an idea which was ridiculous to a whole new understanding, and now Jan is, regarded as an international expert on Venus, gets invited to conferences, all because he didn't believe what Carl Sagan said.
We're going to find out, though. And if it is very different from us, that's a good thing, because that's very likely to be alien.
Yeah, it's more interesting. It's much more
interesting. Now, what do we know from the Bennu samples work? And this is actually a direct listener question. Someone had contacted me and asked this. Mm-hmm. So I'll ask you. Was there any information on chirality? I'm assuming it was both, a mix, or is there anything there regarding that?
Yeah, so the problem that you're having here is very small sample size, and that's basically the short answer to this question, right? You have an extremely small sample size, and to measure chirality in these particular systems is actually a difficult thing to do for sugars. So if I had been, you see, I'm really more of a biochemist than a geologist. So I would have tried to do some enzymatic degradations of these as a way of testing. But they do have amino acids in this, and there is a bias in chirality that's been known for many, many years in carbonaceous chondrites, that is, meteorites have amino acids. No one really knows what the origin of chirality is in these meteorites, but that's one of the things that makes just observation of chirality not a particularly useful biosignature, because it appears that we can get it without life. So the Gil-Levin's experiment, where you couple chirality to a metabolic process, is a much more sophisticated search for chirality. And so the short answer to your question is that there's got to be some more sample. We'd love to go back, with Bennu, this is different from the meteorites. In Bennu, the amino acids were close to 1-to-1 left and right-handed. That is not what has been seen with meteorites that have fallen to Earth, which have been recovered after they fell. There's a huge question about contamination under those circumstances. And so what we're basically saying is that it's up in the air right now.
There have been a few good meteorite falls, carbonaceous chondrite falls like Murchison, where material was picked up immediately, so you've got something. But anytime it's in the atmosphere, it immediately starts altering it.
Well, but Tagish Lake was actually a meteorite that fell into a frozen lake in Canada. Actually, it was much more quickly picked up even than Murchison. But you're right. You try to get to it quickly. And that's one of the reasons why this sample return is so nice, is because we guess that there's no contamination, right?
Now, the testing, though, however, destroys the sample, right? So you get this tiny little sampling of what we only had, just a little over 120 grams of material was what we got. And so you only get this, and then the testing destroys the sample, right?
That's right. So, I mean, in some sense, I guess you could recover some material from the testing, but it's going to be contaminated. But in a word, yes, this testing destroys the sample.
Or at least the scientific value of the sample. You could put it in the Smithsonian or something, but you're pretty much done on the science.
Yeah, that's right.
The science side of it.
That's right. Yeah, no, we would very much would like to have more of it, but you have what you can get. I mean, so keep in mind that Yoshi Furukawa, who has looked for ribose in carbonaceous chondrites, for which we have more, and he has found it there, and he's published this maybe 5 years ago. So it's not a terribly large surprise to find it in Bennu.
Yeah, as I recall, there were 2 carbonaceous chondrites that showed ribose, as I remember.
Yeah,
so there was. I remember that paper. Now, regarding your work with RNA and percolating through volcanic glasses, we talked about this over a year ago now. Any progress?
Has it been that long?
I think it has. Any progress on that front?
Yes. Well, there's a paper about to appear in Proceedings of the National Academy of Sciences, which is under embargo, and so that's going to represent the progress. But you're going to have to come back.
Have to wait for it.
Yes.
Perhaps early in the new year, perhaps.
It may be Christmas Eve.
Yeah, that you don't want.
I hope you don't have any plans.
Yeah. Well, yes, I do, unfortunately. But papers rarely slip by me, especially if they go up on archive. Now, all right, so we have the building blocks of RNA. And this basically says, yeah, Earth had this. You've got everything you need.
You do.
Where does the leap happen in your view once you get the RNA world going? How does DNA come about? Because that's the one thing that was missing is what you need for DNA.
Yes. Well, we divide natural history into 2 things. Okay, before Darwinian evolution and after Darwinian evolution. So if you're trying to put together RNA to get Darwinian evolution started, so this is the RNA first hypothesis or the RNA first model, which is different from the RNA-world model. But if you get life started in an RNA first hypothesis, then you've got Darwinian evolution going, and then you can do anything, right? Everything before Darwinian evolution is constrained by just what is intrinsic chemistry. After Darwinian evolution, you can all of a sudden start doing things that are different. So, I mean, the last, 150 million years, you've seen placental reproduction. Okay, great. 225 million years, what did the dinosaurs do that let them go onto land? Well, the answer is they invented an egg, which had a membrane that would breathe air but not lose water. I mean, when you were an amphibian, you had to go lay your eggs in water because otherwise all the water would dry them out. But the dinosaurs figured out how to make a membrane that would exchange oxygen without losing water. And by the way, that's hard to do. But that's an invention that comes about by Darwinian evolution, which is random variation, at least variation that is random with respect to future outcomes, and then natural selection. And the minute you have that, you can do things that you can't do without Darwinian evolution. So as long as RNA world gets you Darwinian evolution started, then you can start finding an RNA enzyme that will synthesize a protein. And by the way, your modern protein synthesis machinery is RNA. So if I go into the machine that makes proteins, it's ribosome. Now there are ribosomal proteins slapped on the surface of a ribosomal RNA core. And it's the ribosomal RNA core that makes the proteins. And this is one of the archetypal pieces of evidence that you quote in textbooks to say that RNA came before proteins, because in the modern world, RNA makes proteins. Okay, so the other point which we made mention a while ago about how the precursors to DNA are made from precursors to RNA is also, and the enzyme, by the way, that rips an oxygen out to make C5H10O5 into C5H10O4 is a remarkable enzyme, and we've published some papers on it. But, to get that enzyme that rips that oxygen out in the world where there is Darwinian evolution is orders of magnitude easier than to get that process to rip that oxygen out without Darwinian evolution. And the other problem that you have is that once you rip that oxygen out, the deoxyribose is all of a sudden much less stable as a sugar By the way, not as a backbone for a DNA molecule, then it's more stable, but as a sugar, it's much less stable. So you really want to be in a world of Darwinian evolution to manage the process of going from RNA to DNA. Okay, now you do that for the reasons that you've already alluded to, right? It makes the information storage molecule more stable to falling apart. The last thing you need is your hard drive fragmenting, which is what RNA can do, right? It also, RNA folds. You don't want your genetic molecule to fold. Right? It's supposed to lie out there so you can form a double helix with it. So RNA folds because of interactions with the extra oxygen in the molecule. And so by removing that oxygen, you get to DNA, which doesn't fold. It doesn't have anywhere near as many folds. It's a much better informational molecule. Then you go from U, uracil, to T, thymidine in DNA. That adds a methyl group. Well, the AU base pair is not very strong to begin with. Putting a methyl group onto T makes that base pair AT stronger. And so what you're doing is you're looking at 3 steps, right, to improve RNA molecules to make it better suited to be an information storage and transmission molecule, but less able to be catalytically active. So you don't see DNA catalysts really anywhere in modern biology. You see RNA catalysts everywhere, including the molecules that make the proteins. So yeah, so the DNA, the story is, and this is what we teach kids in school. I mean, who knows what's true? The story is that you got the RNA across the boundary from an inanimate world to an animate world, one that had access to Darwinian evolution. Once you have access to Darwinian evolution, the rest is easy, all the way down to becoming intelligent beings and launching rockets to Mars.
Does this lead you to conclude that there is very likely to be life everywhere in the universe?
Yes. Now, the catch there's a great book, maybe, I don't know if you ever interviewed Don Brownlee and Peter Ward. They wrote a book called Rare Earth.
Yes, I have it right up there.
Yeah. But the story is that microbial life, in my view, is everywhere. But the story in that book was that there was a lot of events that were contingent, low probability events, like you having a moon around the Earth, having, the right exact distance, and there's a whole bunch of things that happened that were necessary to get you to the point where you're launching rockets. So you're looking at life, microbial life on Earth, where we have dated this at roughly 4.3 plus or minus 0.05 billion years ago. So to give your listeners a time scale, 4.5 billion years Earth forms, 4.3 billion years, you got life. So that is roughly 10%, well, no, it's 5% of the life of the Earth to date, keeping in mind that the Earth has another billion and a half years before the sun will expand in the main sequence to become a red giant to make this planet uninhabitable. So you've done, we'll call it 2% of the lifetime of the planet, or at least 2% of the planet's habitable lifetime, you got life. But you're 90% of the planet's, or 80% of the planet's lifetime to get intelligent life able to launch rockets. So microbial life is everywhere. Intelligent life, we still have to argue.
Yeah, that may be the bottleneck. My gut feeling, based on all of the science that I've seen, is that, yeah, we probably have a microbial universe. Intelligence, there are problems there. That may not be so common.
Well, you gotta understand what I mean by intelligence. So I got a weird view of this. So keep in mind that you are, as a Darwinian evolution, you make changes in your genome without knowledge of what ones are good or bad. You don't design your genome. And then you have to kill kids, right? Basically, sometimes 90% of the time, 95% of the time, the mutation is damaging, so the kid dies, right? And this is a hell of a way to actually improve yourself. You got to kill a lot of kids just to maintain the information that you already have. So for me, intelligence is to understand your own biology sufficient to escape the Darwinian imperative where you have to have kids die of genetic defects in order to have a pool of mutations able to advance. And if you think about humans right now, we are on the verge of gene therapy where we would be able to make mutations in our genome by design rather than by accident. So we no longer have to have kids dying just to maintain the information you have. And once you have that is the intelligence to escape the constraints of Darwinian evolution. Now you have a mechanism for evolution which is much more efficient than Darwinian evolution. And you already have it, right? I mean, you're wearing clothes, right? You know, that's a cultural information. It's passed from generation to generation. It lets you live all sorts of places on the planet that you would not be able to live. So you're already showing how cultural adaptation, where design is an important part of what you do, allows you to dominate the planet in ways that pure Darwinian organisms don't have a chance.
Yeah, it's true. We are becoming the lords of evolution at this point. And we can, I mean, go so far as to start talking about de-extinction of animals and things like that, obviously nature it has no way to do. You eventually, by pursuing such science, you start saving the kids because you can start to, get rid of genetic diseases and things like that.
I got a good friend who a couple of days ago was diagnosed with pancreatic cancer. 20 years ago, that would have been death. Now, he's up at Langone in the NYU Medical Center, and he will probably survive.
Just catch it early enough still, and in 20 years, it'll be even better.
Yeah.
Especially with things like virotherapy, where attenuated custom viruses attacking cancer cells and things like that. It's a very exciting time for medical science in that regard.
That's right. Well, the only problem is paying for it, and that's, of course, as you know, the most important thing in science is money. No bucks, no Buck Rogers is the line that Tom Wolfe wrote into that movie or the book, The Right Stuff. But it's an expensive undertaking. But frankly, it's in terms of quality of life. My mother will have her 100th birthday in about a week. So I mean.
Hey, they kept my mother. My mother had cancer for 25 years, and they managed to just freeze it, and it wasn't even what got her ultimately. But yeah, she would have been gone long time ago if it hadn't been for advances in medicine.
No, absolutely.
Yeah, absolutely, it's amazing. Now, thoughts on this? So Mars might have life. Okay, we go there, with tourists. Could that life that's there pose any kind of a danger? Or does life, even under very generalized conditions and very unideal conditions based on, what it originally would have evolved under on Mars, can you end up with something very generalized, like a microbe or a virus even, that could pose a danger to the space tourists once they get there?
It's a very good question. Look, I am of two minds on this. Of course, I've already told you I play Ender's Game, so I go back and forth. So on Mondays, Thursdays, and Saturdays, I think Martian life could be dangerous, and on Tuesdays, Wednesdays, and Fridays, I don't. Of course, I take Sundays off, but the bottom line is that it's hard to know. Keep in mind that my number one thing is, I'm holding up, and you have a sample of the Mars as well. Robert Zubrin, who's the head of the Mars Society at one point, made some estimate about 500 kilograms of Mars comes to Earth every year, and we haven't been killed by Martian life yet. Now, then there's a question about sampling. Maybe the rocks that were ejected from Mars and have come to Earth, maybe they did not sample the pond or place on Mars where there is serious poisoning, dangerous pathogens. So that's an issue. The second issue is you look on Earth, the pathogens track with the host. So it's actually hard to find a virus that is not evolutionarily tracking with the organism that it infects. And so it is true that when you have like Middle East respiratory syndrome, Middle East respiratory syndrome probably moved from camels to humans without the assistance of human engineers. The dominant data now say that COVID was actually engineered by people who are interested in understanding how viruses might move from bats to humans. But never mind. The bottom line out of all this is that these viruses, that when they are transferred, cause diseases in the organisms that they've gone into. And so Middle East respiratory syndrome, SARS in 2003, 2004, these were much too pathogenic to travel, so they burned themselves out. So I doubt from general principles that the stuff from Mars, whatever it is, would be parasitic in humans in the same sense that any of the bacteria that have co-evolved with us or the viruses that have co-evolved with us. The viruses just won't. I mean, the virus is very closely attuned to its host. You don't get viruses out of birds into mammals. You know, there's influenza. There's a couple of cases where, but you know, I don't think there's any chance of a virus from Mars. Now, there's a possibility, however, that you would have low-level competition. So they're not competing with you by infecting your cells, but they're competing with you with general resources out there. And so, you know, this is sort of the Burmese python in the Everglades, right? They're eating the grass, which competes with the native alligators. Sometimes they eat the alligators. And so there's a competition at a much less of a pathogen level, unless, of course, you think that the carnivore tiger who's about to eat you is a pathogen. And so the answer is I don't really know, but the level of which we've had so much intercourse back and forth between these species, I mean, that may be uninteresting. That may make Martian life uninteresting, right? We may discover that it was just life that was transported from Earth 3 billion years ago, or it could very well be that Earth life, I mean, I can actually come up with arguments that says that life is easier to generate, to emerge RNA first on Mars than on Earth, because Mars had more dry land where the chemistry that we have is useful in communication with the atmosphere. This is why Io is not a very big candidate for me, because you never had a lot of dry land and communication with an atmosphere that would give you the kind of chemistry that we know makes RNA. But yeah, it's a good question. What's quite clear is right now, technology is available, the science is available to go to Mars and to assess its astrobiological status. What is not available right now is the funds to go do that. So we have been very generously supported by NASA and what's called the NASA Advanced Innovative Concepts Program, NIAC. So they have built for us, we have built for them with their funding, devices that would be a very useful thing to put down with Elon Musk when he sends his water mining operation to mine the near surface ice to make propellant for the return trip. That's an astrobiological sample, which we know how to handle to detect sparse life at the 100 cells per cubic cc level in that sample. And that's got to fly, okay, before astronauts get there. Jan Spacek, the man I mentioned before, has what he calls the impress, as a penetrator thing, right? What's expensive about Mars landing is to land soft, but you can drop 10,000 penetrators off the surface. They may do a little bit of air braking, but you don't have controlled impact, and so you have to harden the equipment there. It will penetrate, but you can land those things somewhere around $10,000 or $20,000, which is something your kids could get a bake sale going, right, in their local high school to actually design a penetrator experiment. So it very much democratizes the exploration of Mars, and that's something that could happen. But the bottom line is that there has got to be funds to do this. Now, what Musk has done is demonstrated these reusable rockets, also now Jeff Bezos and these other people, they basically made the cost of a launch a tenth of what it was beforehand. So you can get a launch for $80 million or something like that. So, but you know, the problem is getting Musk's attention. Of course, getting Musk's attention is difficult under any circumstances, but getting his staff's attention to get them to be persuaded to get onto Mars, the biological detection stuff that they need to have there before humans arrive as part of a robotic mission in preparation for human arrival. This is going to, you know, no one's going to send astronauts to Mars expecting them to wing it to make the fuel to come back without already having the water being mined and already being looked at as a propellant source, for example. But yeah, that's your problem. Your problem is now getting, and the other problem is that sample return. So help me God, that sample with Vivianite, nothing that I would like to have more than have that in somebody's laboratory here on Earth to look at. The problem is that the concern about reverse contamination has made the cost of returning that sample now prohibitive in the current political environment. And so this is a problem. We're sitting here, we know the questions to ask, we know how to answer them. We've gotten enough information about the origins of life, the persistence of life. We have finally broken free of the misinterpretation of the Viking results. But now your problem is actually to get the damn instruments that you need on Mars to actually do the evaluation that you need to have before you send people there. And the Chinese, for all we know, is going to send people there without any of this, though. And that would be a big problem.
It's going to happen. If we lag too much, it's going to happen. It's that simple. Now, what's your view of contaminating Mars? Do we need to take better care not to send our microbes there?
Yeah, I've been on many NASA committees discussing planetary protection, and it's a complicated story, and we are worried about it so much to the point that we don't go to what are called special regions on Mars. And these are actually the regions that are most likely to hold life. So they're defined as regions where Earth life that we know of could perhaps survive on Mars. And so we stay away from those regions because we're not convinced that we can sterilize the rockets in advance of the launch adequately. But yeah, I'm very much worried about it. Again, the environments are sufficiently orthogonal, they're different, so it's hard to imagine what on Earth could go to Mars and not A, be dormant, that is, be able to propagate, and B, to propagate in competition with the organisms that have already evolved to be there. But again, it's very important we have material that goes back and forth between the two worlds. Now, a lot less goes from Earth to Mars than goes from Mars to Earth because the gravity is more here on Earth. The ejection has got to go through a thicker atmosphere, and there's a lot more preventing material to go in that direction. But it has gone in that direction, and so I don't think, and of course, almost anything that you eject from Earth is contaminated with goo, right? It's got stuff on it. So there's almost certainly been transported in that direction in excess of what NASA will deliver. But still, we have to worry about it.
That's actually something I've wondered about, is the material getting blasted off Earth, less than Mars, of course, but it's still out there.
Yeah.
And that might actually be geologically very interesting because there could be some of the earliest rocks that are gone from this planet could be out there still, sitting around on the moon or...
Look, that's exactly right. So people have argued that you go to the moon to do Earth archaeology, right? You have that moon has accreted things on the surface. Now, your problem with the moon is that it is, of course, toasted by high-energy particles. This is why the dust on the moon is shards of glass, basically, that gives you all kinds of problems. But people have suggested going to Phobos or Deimos, right? These are the two small moons of Mars. Maybe they were captured asteroids. There's a lot of discussion about that. But, there's stuff ejected from Mars which lands there. So you don't have a sample return problem anywhere near as hard getting out of Martian surface as you can get off of Deimos and Phobos a lot easier. So maybe that's what our sample return mission should be, and then doing archaeology on Phobos and Deimos. So that's where the humans should land, and that's where they should go to work. And, they won't have a Matt Damon experience, but at least they will be able to do some archaeology.
Well, I wouldn't want the Matt Damon experience anyway. I don't want to be forced to grow a potato on Mars. I would rather do it under planned conditions.
Well, yeah, that movie took a few liberties with events. I mean, the whole premise is that you got blown over in a tornado, basically a Martian hurricane. When the Martian atmosphere is 1% that of Earth, the amount of wind that you get is not all that heavy. But never mind. Yes,
you are going to get a dust devil occasionally, though, that might clean off your solar panels quite frequently. Well, that's how sort of you lost Sojourner, right? The first rovers got too much dust on their solar panels, and therefore had a hard time getting energy. Yeah, but the cleaning events actually prolonged. What was it? Spirit and Yeah, it actually prolonged it.
Yeah,
Prolonged the mission.
absolutely.
A little bit
Well,
tiny.
Yeah,
Yeah.
Well, Sojourner, of course, was the first real try, and they learned a lot.
They did, and that was actually a very neat mission because you could see you had a separate camera on the main landing craft, and you could just see Sojourner sitting there with its mass spectrometer sitting there touching a rock.
Yeah, stuck in the sand. Yeah.
Yeah, I was stuck. Now, regarding Mars, do you think though that all right? So we go to Phobos and Deimos, and we start sending back material from there first. Do you think it's actually, if you're going to do a Martian colony, do you think it's actually more viable to do it in orbit than it is to actually be on the surface?
Look, I mean, I've talked to a lot of young men and women who told me that they want to be the first people to go to Mars, and they don't need to come back. And this is how you go on the Mayflower, right? You pick up and you go. And so Deimos and Phobos is when you want to come back, right? There are no resources by comparison, right? You want to be on the surface where you can get access to the near surface mid-latitude ice. You know it's there because you see scarps that have collapsed where you can see the ice. You've seen it in various Phoenix missions. You've seen it in all sorts of places. So you want that resource for water is indefinite. You need energy, so you got energy and you got water, and now if you got energy, you can electrolyze the water to make oxygen and hydrogen. And then there's something called the Sabatier process, which I'm sure you've heard of, which you take the CO2 and you reduce it with the hydrogen to make methane. And now you can start worrying about using that methane without having to deal with a serious cryo solvent problem. I mean, launching the Saturn with a hydrogen-oxygen propellant was a bear, right? Because that oxygen, you can liquefy oxygen, that's okay, but liquefying hydrogen is worth your life. I mean, it doesn't liquefy to a first approximation. So you're going to have a hard time on Mars liquefying hydrogen, but methane, hey, that's easy. Liquefied.
It's good rocket fuel.
Gas.
Yeah, it's good rocket fuel.
Yeah. Yeah, so yeah, the methane, these Raptor rockets, the methane oxygen combination is a damn nice thing to use. But yeah, so and none of that's available on Phobos or Deimos for anything. So yeah, it's an interesting problem. The question is why you would come back. You do want to, of course, dig yourself a hole. You might want to land near one of these caves. There are lava tube caves all over the place. And, they don't land there as a matter of course now, because if you have a robotic landing and it's, if it tips over or something, you got problems. So they have you've seen these sky hooks and all these incredible ways they try to avoid the risk of an uneven surface landing. But yeah, it's a good question is why you would come back.
Well, I think you've always got a certain type of person that will go and climb Mount Everest. And just because it's there, they go and they climb it. And these are the people that will go to Mars. But they don't build a cabin on top of Everest at the summit. So,
no, that's right.
So there is that.
Well, that's right. And my buddy Chris McKay, he has said to me on many occasions that people underestimate on Mars the fraction of time that you're going to have to spend surviving versus the fraction of the time that you're going to have available to look around. You're on Mars. This is a survival problem, and it's worse than Everest, which is certainly a survival problem. But the only thing you can say about Everest is that if they do the preparation right, you will have a nuclear power plant already built there. You will have a mining operation for water already going, and you don't have to carry everything on your back.
Now, my last question is in regards, we talked about the labeled release experiment extensively, but now the Allen Hills 84001 issue, where we found what appeared to be, I guess you could say, evidence of microbes.
Yes.
Much tinier than our own.
They were a couple hundred angstroms across, yes.
Yeah. Now, what's your view on that one? Do we need to reevaluate that?
Not really. I mean, Joe Kirschvink, who was at Caltech, had a long story about the magnetite particles, the iron oxide in that rock. And he was arguing that there was a certain size and shape of these things that was indicative of a biological signature. The cell-like structures, if you look at them, if you look at them this way, they look like cells. If you look at them this way, they look like mineral crystal spikes and stuff like that. So and of course what's important there was carbonates. There was a lot of evidence of mineral alteration by fluids, and that was in its day very important. I mean, today it's no longer important because we have a lot more direct evidence. And then Allen Hills is also very important because it really is one of the oldest rocks we have anywhere, okay, in the solar system. But the ultimate reason why Allen Hills was important is because it brought Bill Clinton to the Rose Garden for an address and showed the public's interest in this problem. And that Dan Golden, who was the head of NASA at the time, he's still living as far as I know, he was a good enough administrator to turn, the question, is this life or isn't it, which is what Bill Clinton allegedly asked him. And he said, allegedly, I don't know. And then Clinton allegedly said, what the hell are we paying you for? I mean, when the little green men invade, you're the guy I'm going to go to. But Golden was able to leverage that into the 20-year program of astrobiology, which is, frankly, a jewel in American science that has managed to fundamentally transform how we look at space exploration with respect to life. And the reason I can sit here and have all these discussions with you about borate and about Vivianite and all the rest of it is because Golden was able to understand, and Clinton himself, and of course Congress as well, was able to understand the public interest in this to manage to maintain it. And, it was not a lot of money, but, it's maybe maybe $100 for every man, woman, and child on Earth. No, I beg your pardon. That would be just in the United States. Maybe about $25 billion. $25 million. I'm sorry. I'm sorry. I'm overestimating that by orders of magnitude. It's not a lot of money. It was $25 million for the astrobiology program compared to launching humans into space. Of course, that's a lot more expensive because you're not allowed to kill the people. But yeah, so it was not a lot of money compared to the NASA budget. Maybe it's 10 cents per person. But frankly, it has taught us much more about this problem than I ever thought. I mean, I didn't think 20 years ago I would be sitting here telling you we have a good idea of how RNA emerged on rocky worlds. I wouldn't have said to you, if you asked me how abundant microbial life is in the galaxy, I would have had to tell you just I have no idea. And that's no longer the case.
Well, we learn more as time goes on, but I do have to say that what I remember of Dan Golden was that man was a fantastic public speaker and a good salesman.
Yep.
But he could definitely put the wonder into what he was talking about regarding NASA.
Well, that's his job, right?
Yeah.
I mean,
he was better than average even for a NASA administrator, though. Well, he was very good.
You asked Ronald Reagan whether he thought he was, as an actor, he was well trained for the presidency, and Reagan will respond to you, I know I'll get in trouble for saying this, but frankly, I don't see how you could do this job if you were not an actor, right? This is the job. So Golden's job is to be able to explain to lay people, especially taxpayers, why their money gives them a sense of purpose or a sense of mission. I mean, this is what NASA sells the public. If you're lowbrow, you go watch the World Wrestling Federation, right? If you're highbrow, you go look at Hubble or Webb telescope pictures. So what NASA provides to the American public is a sense of mission. It's inspiration for kids to go into science and technology, right? Nobody will go and suffer through science and technology. It's hard work, right? But Golden was very good, and of course NASA is very, very good in motivating people. So almost everybody I know here who comes to my laboratory is motivated to go into science in some sense or other by a space exploration mission. And mean, Steven Spielberg can barely hardly make a movie without an alien in it somewhere, right? I mean, it just has captured the imagination of the American culture, and that's, of course, one of the reasons why the American culture is so science and technology oriented.
It's true. It's true, and NASA needs more funding. I'll just put it that way, because we're going to need to answer questions like this Mars life question. And now with the building blocks being so common apparently everywhere in the solar system, that's we need to do more. All right, Dr. Bennu, thank you for joining us today. Tell everybody where they can find your blog.
Well, we have a blog called Primordial Scoop, which is like primordial soup, but it's SCOOP, and it's .org, not .com. But we also have the Foundation for Applied Molecular Evolution, which is www.ffame.org as well. So you can go see what we do. Now, you got to keep in mind that most of what we do at the Foundation is medical research to cure cancer, diagnose infectious disease, to manage what happens the next time a COVID comes along. We had, within 8 weeks, we actually had a COVID test ready, and it was approved in India in April, actually. So this is what we really do. So the exploration of space is a relatively small fraction of what we do, but that's where you can find me.
All right, thank you, Doctor. And I look forward to the upcoming paper under embargo.
Oh, you'll be the first to know about it. You'll be the third, maybe the 15th to know about it.
You bet. And I hope you'll come back for another round once that's out.